My name's Jacob Oster. I'm a master's student here at the Chesapeake Biological Lab. I work with Laura Lapham and Andrew Hayes, and together we're looking at mercury pathways in aquatic ecosystems. I'm looking at aquatic insect communities in streams, and what we want to understand is how these organisms absorb mercury into their bodies, and then depending on what organisms eat these ones, understand how mercury absorbs and concentrates as you go up the food chain, and then which organisms then could be a potential risk for high mercury concentrations leading to mercury poisoning. Mercury is a neurotoxin and it transfers across the placenta very easily, so it's especially a concern for pregnant women. They don't want to eat fish that have high levels of mercury because then it could potentially harm the unborn child. We're contrasting two streams in the Kilpatrick Marsh watershed. One of them has a high history of agricultural use. The other site's been largely untouched by agriculture within the past 100 years. When I go out into the field, I collect water samples, I collect sediment samples, and I collect insects throughout the stream. So once I get back to the lab, for the water and the sediment samples, I analyze them for total mercury. For the insects, what I have to do is I have to identify them to the lowest possible level I can. Usually this is the family, genus, or species level, depending on the organism. So some of the more common insects that we see in our streams, we have caddisflies, we have Helgramites or Dobson flies, we have some small species of clams, we also have some species of worms, we have these crustaceans, amphipods and isopods. What we can do is start looking at the different dynamics within the community. Are these organisms that mostly remain within the sediment? Are these ones that feed on leaves that fall into the stream? Are they predatory macroinvertebrates? Which organisms then may be tied to the mercury as it enters into the stream? So what we're finding is that it's actually the largely untouched watershed that's contributing more mercury. Our goal then is to tie this into Maryland's Biological Stream Survey and give them another tool to look at what could potentially be going on in their streams. And if they see communities that are similar to ours, this could give them an idea of like, okay, this stream should be relatively safe for mercury consumption. If we're seeing these other insects and they're present throughout the survey time, maybe looking at the concentrations in those insects to help understand what's going on in the fish.